I would love for you to join in with me as we dive into some DNA and some protein synthesis, which begins there on page 94 in your lovely texty book. Shh. Jump in quickly. No need to converse. It's always one of those funny things as a teacher. Whenever you tell someone to take out their books, somehow the talking begins. It's a weird thing, Mikey, like always. Not Mikey, of course. He didn't do that. But. All right. So we're diving into those nucleic acids. That was immortalized forever on. Yeah, I apologize. Because you weren't talking at all. I just picked you out because you were looking at me. All right. Nucleic acids and protein synthesis. All right. You remember last unit, we were talking about the different types of organic molecules, and nucleic acids and proteins were two of them, right? What were the other two? Do you remember? Carbohydrates, good, and lipids, good, excellent. Proteins, lipids, carbohydrates, nucleic acids, right? Those are our four main categories of organic molecules. So DNA is basically the holder of our genes. Okay, well, what is genes? The genes are the DNA information, the genetic information that makes you, you. It's fine, it's fine, it's fine. And we need that information to actually make a protein. And we'll get to that in a moment. But we'll talk about the DNA here first. So DNA's got three pieces to it. You've got a sugar base, and then a phosphate that's attached to that. They call that the sugar phosphate backbone. And then you have this little base pair that extends into the interior. For DNA, this sugar, the name of it is what? Anybody? It's the D. Deoxyribose, right? And if you were doing an RNA, it would just be ribose. Deoxy means no oxygen, right? So it's very similar. The sugar molecules are very similar. It's just one has oxygen in it, the other does not. Okay? Or, well, we'll leave it at that. The phosphate pretty much stays the same. And then the bases are what change. So you remember that these bases get paired together, right? That there are four of these bases. And again, you've heard this like 1,000 times, right? I know you have. Come on, Eddie. That we have thiamine and adenine and cytosine and guanine. And these guys pair together, right? So easy ways to remember what go together. TA, Tacoma Academy, or AT, Appalachian Trail, that's how I remember it. GC, the General Conference, or CG. I don't know of any CGs. Would you all know CG? Like what? CG. It's close. CC. Caramel and granola. Perfect. Whatever you remember. But knowing that that's how these guys pair together. So if we look at that DNA piece, you see that C is always paired with G. G is always paired with C. A is always paired with T. T is always paired with A. That's how it works. And that simple code, isn't that a little bit crazy to think of? That simple code of these pairs, in whatever order they're in, that is what gives all of the instructions to build every cell in your entire body. With all the variations, with all the extra pieces that needs, all the adaptation pieces. It's pretty impressive, if you ask me. It's all contained in that. Of course, you know that DNA is a double helix, right? It spins. So the blue pieces, that would be the sugar and the phosphate backbone, right? And then these guys in the middle are your base pairs that are paired together. How does DNA replicate itself? It's actually kind of cool. It's like a zipper. Inside that nucleus, that DNA kind of unzips it splits apart slightly. So that instead of having one piece together, you kind of have two separate pieces in just a little spot. Not the whole DNA unzips, just a little piece of it unzips. And then you have these little base pairs that are brought into the nucleus. So these little bases, these guys right here. Bases mean this is the base, right? Remember we had the phosphate and the sugar, the blue part? And then the C, T, A, G, those are the bases. And they're little just kind of random ones of these that are out there. Kind of like your little Lego blocks, right? And they match up. The C's with the G's, the T's with the A's with the T's. And they literally take and make a whole new strand of DNA. They actually make two strands because one side was the original and the other is the side that has now been created as the DNA replicates itself. And just in the last few years, we've learned that there's a whole process behind making sure that that doesn't mess up. Because you're comfortable, there's an awful lot of base pairs there, awful lot of codes that's there, and if you mess up just a little bit of that code, well, now that protein no longer works. Now that part of the body doesn't function the way it's supposed to. So there's a whole huge system that helps keep that in the right way. Yes, ma'am?
In the last slide, what did it say about the second like the one before.
Here?
The last slide.
Sorry.
Yeah.
It's just the double helix means that twisting form. So rather than just being together, it's double meaning there's two sides of it, right? Two of these sugar phosphate backbones that kind of just twist around each other. And that's the name of the shape, a double helix.
So the strand itself is the phosphate?
Right, the phosphate and the sugar is the blue part. Yep. And then the bases are just these guys in the middle. Okay. Are you with me on this part? It's simply that the DNA unzips. I love that kind of thought. In my mind, that works very well. If you think of a zipper being together, it just literally unzips that zipper and then copies on both sides of the zipper to make two totally new DNA strands. And then, of course, remember from last unit, one of those strands will be transferred over into a new cell when the cell replicates. I'm sorry?
Does it break apart?
Eventually, you would have two separate DNAs, absolutely.
And it never comes back together?
No, because it's just an exact copy. Yes, ma'am.
So is the DNA like, does it only unzip like at the ends?
Right, and then it would slowly work its way up. You got it. You got it. There is other unzipping that takes place. We'll talk about that in a second as you're making new proteins. But we'll talk about that in a second. But as far as replication goes, yes. On an end, unzipping, and then copying both sides of it. That's a great question. I don't think so. Yeah, I think it can go from either side. Yeah. Ooh, almost falling. Okay. That genetic code is the sequence of these bases, right? So that code tells the cells what to do. It's information, right? It's how the DNA stores information, is the sequence of these bases. We have things in our cells called RNA, again, the ribose nucleic acids, that their job is to come in and copy the information from that DNA and then take it outside the cell in order to make the proteins. Where are the proteins made, by the way? What part of the cell? The ribosomes, right? So we just talked about mitochondria, how they worked, and now we're diving into these ribosomes. The RNAs have a little bit of a difference. The G's and the C's are still there. The A is still there. But instead of the thymine, we have uracil. So it is a U that goes together, which of course stands for Andrews University. The place that if you want to go to heaven, you need to go to Andrews. That's just the way, that's just how it works, right? No, I'm joking. All right. So there's two types of RNAs that we're going to learn about. One is called messenger RNAs. The other is called transfer RNAs. So the messengers are the ones that actually come into the nucleus. They're actually made in the nucleus there. And they copy off a bit of the DNA that we call that process transcription. Have you heard of a transcriptionist before? Where do they work? Sorry? Oh, no, don't know. Okay. Transcriptionist. They're typing, sure. Sure, right. So from a video, they were the ones who are actually doing the typing of the words, what the people are saying. In a courtroom, these are the folks who are keeping track of literally every word that's uttered, right? In a doctor's office, I used to, when I worked at this one PT clinic, we would dictate our notes. Instead of actually writing them, we would just talk into a phone and dictate, and somebody was transcribing what I was saying, and that was my notes. It was actually kind of nice. I didn't have to write, because you all know how terrible my handwriting is. Your question?
Oh, but like, if you're in a courtroom and like you get to sign on the writing,
You can tell them to pause, but that they don't like that very much.
so they use shorthand.
They do. It's a whole process. Those ladies and gentlemen, mostly ladies, who are rolling, man, because they type so fast. It's a special machine. It's not actually a computer. It's a special type of keyboard they use to use that shorthand. But literally every single sound and word that's spoken, they're recording. They do. Yeah. It's pretty impressive. Okay. I'm sorry?
There's like a fight in the courtroom. Is there
I'm assume they are keeping track of what's being said, yeah. Yeah. They're showing you here, this is the pairings. Shh. Over here you have a DNA. Over here you have an RNA. Noticing that the A from the DNA is not paired with a T as it normally would for a DNA. It's paired with a U, right? The T here is still paired with an A in an RNA. That's fine. The G's and the C's and the G's, those still stay the same. But again, an A for the DNA pairs with a U in the RNA. That's one of those clear differences that people always like to test you over. What's the difference between RNA and DNA, right? That DNA has the T's, RNA has the U's. These are the bases. The bases, right? The adenine, the thymine, the guanine, the cytosine. These guys are the ones that are the bases. So each DNA and RNA is made up of a sugar, again, ribose or deoxyribose. The phosphate group that's attached to that, those make that blue backbone. This piece right here, the blue part, that's what the sugar and the phosphate. And then these guys are the bases. Sir?
So I thought like DNA is its own strand and RNA is its own strand.
They are. What they're showing you is this is the process of where this is going to copy a little piece of that. So yes, they've kind of taken, unzipped just a portion of the DNA to get to a specific spot that has the code for a certain protein. We're going to talk about that right now, how that works. Okay? What page is this on in your textbook for me? Page 98. This diagram has got a ton of good information on it. So let's spend some time here. So inside the nucleus, that's the big blue thing on the left, right? On page 98, right? Good. Inside the nucleus is where the DNA is. Remember, the DNA contains what? The DNA contains what? The code, the genetic code, which is what? All the instructions to make the proteins, right, that make up all the cells that do all the things in your body, right? Think of the DNA as a cookbook, all right? But this cookbook is so valuable that it's protected like in this most holy place. Like the old school medieval libraries that used to chain their books to the shelves, right? That's what this is like. This code, so maybe y'all didn't know that was the case. Because Because back in the day, books were super valuable because we didn't have this little thing in our pocket that had, yeah, sure. All right, so, oh, right, sure, there you go. Dr. Strange did have that in there. You're right. Good. So, inside that nucleus, when the cell, talk about Dr. Strange, y'all get rolling. I like it. When the cell wants to copy a protein, when it wants to make a protein, it has to go inside the nucleus and get the instructions to make that. What does that are these messenger RNAs. Messenger RNAs are literally assembled in the nucleus from the code. So this diagram I'm showing you here that has the pairs together, these guys would come and form, and they start making a chain of whatever that code is. Maybe the code is only a few base pairs long. Maybe it's hundreds of thousands of instructions that they need to make a certain protein. Are you with me? So it's like sending in one of the junior chefs into that most holy place. You can't take the recipe book out, but you need to copy down what the recipe is. Or maybe your mom has a recipe book that has all the recipes she does, and it's like she guards it closely. And okay, you can copy it down, but you can't borrow that book. I need it. We can't have anything happen to it. Maybe not. You with me? So that messenger RNA copies the information from one half of the DNA, which of course is just the mirror image of the other side. And it takes that information out of the nucleus. That's what I'm saying, the opposite of it. Yes. Are you all with me here? The mRNA goes out of the cell now. And it's carrying that code, right? It has the code of how to make that certain protein. It takes that code where? To the ribosome. Why? Because that's where the proteins are made, right? So make sure you're connecting what you've learned in the past to what you have now. We said we know the ribosome is where the proteins are created, right? Protein synthesis, that's what it does. This is the process of how that works. So that messenger RNA, the mRNA, comes to the ribosome. And they begin a process, let me go back here. Oh my. We begin a process that we call translation. So transcription was in the nucleus copying down the code. Right? I'm transcribing it. I'm writing it down. And then I come out to the ribosome, and now I'm going to translate. Let's take this code and translate it into proteins. Even more specifically, into amino acids. What we find out is that for every 3 of these bases, that equals a certain kind of amino acid. So the code AUG, we call that a codon, those 3 bases. That codon is equal to this methionine, which is a specific amino acid. It's like letters. These amino acids are like letters. Like if I wanted to have you write a word, I have to put these letters in a specific order, right? To spell Nathan, N-A-T-H-A-N, right? And each of these 3 base code is for a specific letter. Am I making sense to you? A couple of you at least. So, inside that cytoplasm, a second RNA, which is called transfer RNA, has what we call an anticodon. It's the bases that fit on to the codon. And they bring those and they match them up on the ribosome. Here's my picture. So here's my messenger RNA, right? Here is the 3 base codon. AUG is a codon. GGC, that's a codon. UCC, that's a codon. GCA. Are you with me? Hang on one second, I'll get your question in a second. And on each of these little transfer RNAs, they have another codon. But we don't call it a codon, we call it an anticodon, because it's the opposite of the codon. So see how this is an ACG for number 5 here? The anticodon would be UGC, the pairs that would match up, right? AU, GC, they'll match up, right? And when they do that, they bring and they say, okay, this is my spot here. There's an ACG, I'm a UGC, boom, I'm going to meet up those two guys together. Are you with me? Each of these are carrying with them an amino acid. Oh, come on now. An amino acid. We're trying to line up these amino acids in a nice, specific chained order. Some questions? Please. This is translation.
And you said the amino acid would be like a number 5. Or is it the whole chain?
No, each of these little guys, 1 is an amino acid, 2 is an amino acid, 3 is an amino acid.
So what would you call it?
It's called a polypeptide chain.
Is it like a
It's the beginning of a protein. Remember, we then have to go through conformation where we take and we bend and shape this 3-dimensionally to actually do what we want it to do. Okay? Sir?
So does the amino acid come already connected to a specific
Anticodon. Yes.
So there's just anticodons floating around.
In the cytosol. Yep.
That it's going to match up. That one comes.
Exactly.
Okay, so it's
Exactly.
Not like it's like making it.
No. These are already together. They're already, when you have eaten the proteins that their body has broken down into these smaller amino acids, they are now hooked onto these transfer RNAs to allow them to. I'm super simplifying, but yeah, they're already present in that ribosome, in that cytosol area.
Are they all just there?
It's the transfer RNA. Like, that is what it is. So it's another protein that's been created by the cell that matches up with these amino acids. It's basically like the matchmaker who's going and saying, oh, okay, Nels, here, I've got somebody for you. Let me come bring you over, and boom. Here. Does that make sense?
So then, like, when, as the messenger RNA is going to make the code, is it almost like an order, like, it tells the ribosome, like, hey, these are all coming, so we need to...
Right, sure. It's making a certain protein. That's what this messenger RNA is. It's saying, hey, here's the code for the protein we want.
No, I'm saying when the messenger RNA gets copied, does it say, like, does it tell, like, the body what anticodons it needs made?
You've kind of just got a whole slew of them that are around these ribosomes. So it has what it needs to do, what it needs to be done. Yeah, it's a good question. Ryan, did we get you?
For the anticodon for the, like, an A is always going to be an E.
Yes. And again, the anticodon is a 3-base set. So it's not just the U and the A together. It's actually these 3 that make a codon or an anticodon. The codons are on the messenger RNA, the anticodons on the transfer RNA. It's good. Well, on the messenger RNA, there are no T's. So there's not going to be any T's involved in the transfer RNAs. Does that make sense? Because again, you're doing the GC, the AU with the RNAs. The T's are just with the DNA. You got it. Are you with me, my friends? So they line these guys up, right? So here, number 5 and number 6 have been lined up together. And then between them, between these amino acids, there's a bond that forms, what we call a peptide bond. And that bond is what holds those 2 amino acids together. So there's a bond between number 1 and 2, a bond between number 2 and 3, a bond between number 3 and 4, etc., etc., etc. Are you with me? Once that peptide bond has been created, then the transfer RNA will break off of the messenger RNA. It'll go out and find another amino acid that it matches, another number 5, and it bind it and bring it back to the ribosome so that we can keep making these proteins. So it's basically the worker who is going to get the parts we need to make this specific ribosome.
So it's like a number 5 and it's like hopping around.
Yeah. Sure, it's just kind of, because we've eaten this protein that our body has broken down, send it into the cells. Now the transfer RNA will go and grab that specific protein, that specific number 5. Again, they all have names. You don't have to learn them all. But each of them have a name, a specific anticodon that goes with that amino acid. Yes, ma'am? It'll collect them and bring them to the ribosome where the messenger RNA is present. They are just in the cytoplasm, yes. And their job is to bind onto those amino acids, that one specific amino acid it goes to take it to the ribosome, to make these proteins. And once that number 5 has been connected to number 6, the transfer RNA goes away, and now we do the same thing here with number 7.
So the transfer RNA already comes with the amino acid?
Always, yep. Yep, and it goes back and then gets another one and comes back to the ribosome. Yep.
Oh, so there's like one of each transfer RNA. It's not like there's...
Oh, there's plenty. There's tons of them. The specific is that this goes with number 6. No, there's lots of them. There's lots of them present.
So then once it brings an amino acid, can it do another one, or is that like...
It'll go off and get another one and come back, sure. Yeah. Mm-hmm. Are you with me, my friends? I think that's actually the end of my slideshow there. And again, we call this process protein synthesis. So we'll end up with a chain of amino acids. Maybe it's 10 amino acids long, maybe it's 20, maybe it's 100,000. That's an awful lot, but it could be. And then that chain now has to go and do conformation, right? Go through that process of molding and bending and shaping itself three-dimensionally to fit into the keyhole, right? To fit into whatever function it's supposed to be forming. Any questions there? Perfect. Perfect. What I want you to do then, we've got about 9 minutes remaining in this class. I want you, to the person next to you, explain this process. Together. Explain this process of what's happening in the ribosome in this protein synthesis.